Yeah, so in this talk, what I wanted to do is talk a little bit about visualizing the cell simulations that we've been talking about with Tyler, how you construct the geometries, how you build your system. And today I just want to tell you about how we can visualize that using the popular molecular dynamics software, VMD, that is also developed here at UIUC in the TCPG. And so to kind of give you an outline of what we're going to talk about today, we're going to have the overall goal, overall motivation of learning about how to visualize lattice microwave cell simulations in VMD. And to kind of get to that point, we're going to have to go through basics of just using VMD for people who might not be familiar with that. We'll give an example of the molecular work that people typically do if they're doing atomistic scale simulations. So maybe looking at how a protein binds to another protein, or transport of water through the membrane potentially. And then we'll finally move to actually looking at cell simulation, not only visualizing the geometry, but then making a movie to show the dynamics that are occurring within our reaction diffusion system. And finally, just cap it off with some takeaways of maybe important commands for you to know and new additions to a plugin that Tyler's developed that will be useful for you going forward if you're using lattice microwaves. And so we've seen this slide before. It's a typical workflow that we use for lattice microwave simulation. And so we can see the left here, the experimental data that we usually take and build into our simulations. Then we can use Jupyter notebooks to kind of input and refine those simulations that like we've done in earlier tutorials, and then run through AWS cloud computing, like we've been doing throughout this course of this workshop, on a local machine or on a supercomputing cluster. And so the point we're really going to focus on now is after I run my simulations, how do I visualize the results, not only the geometry, but the dynamics of the system. And then we can kind of continuously go through this loop, maybe refine the geometry in some way and make some changes to our reaction system and continuously see the results. And so the first thing we're going to go through that's important to note if you're not familiar with VMD is there's a certain syntax for selecting a different substructure of the molecule that you might be looking at. So you can select an entire protein, for example, a specific amino acid or residues by positions of the residues or by some type of secondary structure. And there are tutorials for this available on the TCPG website as well. And so with that, the first live demo that we're gonna start off with, and hopefully it'll go pretty well, is a demo of looking at the structure hemoglobin that's used to help provide and carry oxygen throughout the bloodstream. Fire this up in VMD real quickly. So just open up the newest version of VMD, and which is also interesting to note, there's a quantum chemistry implementation in that. If you're interested in doing that type of simulation, that is available in this new 1.94 alpha version. And so what we typically do in VMD is we can load a molecule. In this case, we would just download a PDB structure. So just go to pdb.com, and that's not what we're looking for. Just Google PDB, actually. Not. This is going well already, right? There had to be there we go. There had to be a little comedy portion of this talk. Okay. And then it's nice if you can look up the structure of your molecule, but I'll just type in hemoglobin here, and you will see what comes up. I don't actually want that one. I want this one, human, because humans are pretty cool. So we'll do that. And then what you can do is just download the file in PDB format, and that can be read by our VMD program. So I've already done that. So we'll go back here and just put browse, go to where we saved it in our directory structure. And now we will enter the wonderful world of VMD. And so here is our protein in a typical ball and stick type structure. Hopefully the lighting's good enough there that we can see that for everybody. Okay. And what we can do then is you can go to graphics and representations. And this is kind of the toolbar that you're going to use to select different parts of this protein and make changes to the appearance and maybe the color of the different portions. And so right now we have the entire protein selected, but what we can do that might be more interesting is just look at one of the kind of key constituent parts of hemoglobin, which is the iron. That's where the oxygen is going to get bound and carried throughout the bloodstream. So if you want to do that, what we have to use is some selection keywords. And that's kind of the basic structure of VMD, how you would select different parts of these macromolecules. So you just go to where it says selections over here and click on that. And then you can see there are different keywords, either based on name or the residue type. And so one of the residue types here is protein. So if we select that, it will select all of the protein residues. But if we want to see iron, what we're actually going to select is not protein. And then you can also select the atom. So Fe. Let's select right here. Let's just check to make sure and name Fe. There we go. That's important to do. So yeah, like I said, one of the keywords is name, so important to type that in, which I just illustrated, not on purpose. I wish it was. That would have been more educational, but so not protein and name Fe. And then so right now we can't see anything, and you're like, wow, that's not good. But it sort of makes sense because for an atom, if I want to draw it as a line, that might not be the best representation. So instead, if we can look at it in the van der Waals representation, so there are 4 iron atoms. And now that we have those, what might be a little bit more interesting to see is kind of the substructure around that, so the heme groups themselves. So what we can do is create a new representation. And this time, what we'd like to see is a heme group. So just type in heme. And we will try to make it a different color, because that will be useful for us. Can't change the color before we actually make the selection. So heme, and we'll change its color so it's not the same color as the iron. So the heme groups. And this time we will actually make this as a surface because that will be better for us to look at here. And the only problem is we've kind of lost what the iron looks like, so we kind of want to blow that up in size a little bit so we can still see it within the heme. So now we have our iron in our heme group. And then the final thing that might be interesting is to see its position within the entire protein. So we can then just select the entire protein, and we will do this looking in the kind of familiar chain cartoon structure, and we'll color it so that the different subunits are different colors. So just go with the chain, and now the subunits are different colors. You can see those. change the color of the heme, so that will be different than the chains. And so now what you can see is 4 different chains of your heme group. You can see the iron atoms, and you can see the different heme groups. And so that's a typical example about how we'd make selections in a molecular picture. So that's good, but what I'd like to do now is move more into how we would do this for a cellular type picture. And so we'll just go back here. Okay. So we're trying to visualize the entire cell, like what we see in this picture. This is a yeast cell. And there's a few differences with working with the lattice microfiles in VMD as opposed to working with a PDB file, like what we just did. So what we can select now, instead of maybe different atom types or different protein residue types, is the different cellular regions or compartments, such as the membrane or the cytoplasm. We can actually view the lattice geometry that's used in the simulation. So maybe instead of residues, here we'll actually look at the voxels or subvolumes that were used for the RDME simulation. And we can also view specific species, maybe specific proteins or mRNA in our simulation system that we're interested in seeing the dynamics for. And so the example that we're going to look at here is the MIN system or MIN-D system, which I think some people in the audience are fairly familiar with this reaction diffusion system. And so the key role that this plays in E. coli is kind of determining the location for where cell division should occur via a network of different proteins that bind to the membrane. And so 2 of these proteins, MIN-D and MIN-E, will oscillate between the cellular poles and kind of prevent another protein that is FTSZ, FTSZ, from binding at the ends of the cell and eventually localize it towards the center of the cell so that when this cleavage happens, it happens in a uniform and equal manner. And so what's really interesting about this from a reaction diffusion standpoint is that we'll view these 2 protein species oscillating between the poles of the cell, and that we can pretty well define the reaction diffusion system in terms of the MIN-D and MIN-E proteins binding together, getting activated via ATP, and then diffusing from the cytoplasm to the membrane. So it's a pretty simple reaction diffusion system of only about on the order of like 10 reactions. And it's been fairly well studied in the literature as well if you're interested in learning more about that. Okay, so we will now go and do a live demo of this in VMD. And so what's really nice is, in a similar manner to what we did with the protein, you can just load your completed simulation files into VMD in the exact same way. So we just go to New Molecule again, and now instead of looking for that PDB file, I can just load my .lm file, just like the files that we all created yesterday in the tutorials. So we'll just browse for that. Have our MIN system file here, so we will open that up. And one thing that's kind of important to remember here is now we have data. Our protein data before was stationary, right? That was just the protein in one state that was maybe captured by an experimental technique like X-ray crystallography. But here in our simulation data, we have dynamic data. That is the state of the cell over many different time steps. And so what you can do is, if you look in this region over here, you can actually select a stride that you want to take in loading the data. So MIN-D, I think, is typically found to oscillate on the order of like 30 seconds from one end of the cell to the other. So I've simulated for 600 seconds. And if we didn't want to see the data in such a refined format, we could put a number like 10 in this stride box. And so kind of using the indexing that you might be familiar with from computer programming of an array, the first index starts at 0, the last index is equal to -1, and this just means that we'll skip every 10 frames. So every 10 time points that were saved in our simulation file, if we were saving on the order of 0.5 seconds, I won't see the data every 0.5 seconds, but I'll see it every 5 seconds. So that's something we could do. For this type of simulation that I did, I didn't do a very long time, and I think it's nice to kind of have the time resolution. So we'll actually just keep it at 1 here. And then you can just click load, and here is our full ISO. And so you can see here, it's loading all the frames.
I have a question. Can you also use this option of, you go to the load window, you have, can you use, when you still load, I think it's just behind it.
Yes.
Can you still use this load all at once option?
Can you still use the load?
Yeah, because normally when you have an empty trajectory, you know,
Sure.
I mean, you don't want to have this loading step by step. You can just load the whole thing and wait for the trajectory to be loaded, and then you have it. Sometimes, like, I mean, like this.
Oh, it could take like a very long time. Yeah. So you
can use it.
Still use that feature. Yeah. Yes. And so if we just rotate a little bit here, just using the mouse, we can see kind of what the E. coli cell looks like a little bit, with this blue representing the cytoplasm, and the pink representing the outer part of the membrane. And if we go to here, our graphics toolbar and representations again, we can start making selections of the molecule. And what's interesting to note here is if I go to the selections window, we can see actually the keywords are a little bit different. So before they were the different atoms, different residues of the protein, but now we can see instead of that, it's the different cellular regions, so the cytoplasm and the membrane, and then our different RDME species, the VMD protein and mini protein. And so we can start making selections of these kind of units instead of doing it from an atomic picture. And so for regions, what you can see that would be useful for us, and actually scroll down so we can just see it in here, is the seg name. You can select 2 different options. One is site, and so site here, that's going to correspond to the regions, so the cytoplasm and the membrane. And then the other is RDME, and that's how we select the RDME particles. And so first, we will just select the cytoplasm. So seg name site, name cytoplasm, and the membrane goes away. And we can think of a nice way to color this. So that it will look good in our simulation. Okay, so we have just the cytoplasm right now. And what we could do next is select the membrane. So we'll create a new representation for that. And what might be interesting for the membrane is to see it instead of as points, instead of as different sites in a point formation, is to see it as a surface. So you can zoom in and kind of see that structure. And another interesting tool that the people who use VMD and might be familiar with is you can actually create, sorry, let's see if I can pull it up here. There we go. By hitting the number 2, I guess you can't really see the mouse now because I'm using a black background, but by hitting the number 2, you can actually measure distances as well. So if you wanted to see what is the diameter from top to bottom across this capsule, you could click in one position. Maybe I'm a little too high here. It is 2, right?
Yes, you are correct.
Okay, well, generally you can click in one position and then another position.
Oh, with the surface, at least my experience with the surface representation, and it with proteins.
I see, that is actually the issue. You are correct. Going in the wrong order will make problems happen. So let's not do a surface. Let's do points. There we go. That's a little bit better. It's actually good too, because you can kind of get an idea of what the lattice spacing is now for our RDME simulation. But yeah, so let's do points. And if I do that again, just wait a second for it to come up here. I'm in the selection bar, so that's what's happening. If we do that again, then you can select a distance from top to bottom of the cell. it will actually measure the difference. And so here you see this value, this number. And so in VMD, the distances are measured in angstroms. I didn't click exactly at the top and the bottom, so it looks like it's about 8,600. It should be about 10,000 because this is a 1 micron diameter E. coli cell. And so kind of with that in mind, what we could do now, zoom out a little bit and get our selection window back here. Is you could cut off the membrane at some point. So we could say y less than 5,000, and now you will see the top half of cytoplasm exposed. I think we have a question here.
Can you also use other programs like Chimera, or is that the only program which visualizes the cell?
We do not have a plugin.
Yeah, so there's no support for lattice microbes in Chimera. And so yeah, you could view maybe the proteins in a similar manner, but we don't have support for that for lattice microbes. So yeah. Okay, but for seeing the molecules in the reaction diffusion system, looking at the membrane actually probably won't be that interesting. We'd like to see more of the dynamics within the cell, movement to the membrane. And so then what we can do to do that is create a new representation where instead of looking at a site now, we will look at the particles. And so we'll do that again by doing selecting the RDME option instead of the site option. And we can select the name of one of the proteins, so MinE, for example. And now you can see in these little red dots where MinE is, but we'll make those a little bit bigger, I think, just so we can see them. Might actually be a little bit easier to see here. There we go, not too bad. Okay, so then we can see where our MinE is located within the cell, and we can also do this for the MinD proteins that will be oscillating throughout the cell. So we'll create another representation to do that. And the nice thing with VMD as well is you could select 2 different proteins in one type of selection if you're not really concerned with the difference between them. And so for the 2 different variations of MinD, either bound to MinE or unbound to MinE at the membrane, we might not really be concerned with the difference in terms of watching the oscillation. And so just by using a logical operator like OR, we can make that selection. And we will choose a different color so things don't get too confusing here. Purple, was that nice? Yeah. Okay, so red was not a good choice because that kind of doesn't go very well with purple. Green, good audience suggestions, I guess. Green was also, see, there we go, teamwork. So green, okay, so now we can see our different proteins that within our system that will be interesting for us, and we'll just zoom out a little bit here. And so what we can do now is actually look at a movie, potentially, and see the dynamics of this system. And so if you want to do this the sort of lazy amateur way, because I'm not really an expert at VMD, we can go back to the initial frame of the simulation where we only have these MinD proteins. And just by pressing this play button, it will go through all the frames of our simulation. So here we go. And you can see the MinE protein number kind of start to decrease. And as we increase in time, if I speed this up a little bit, we'll start to see the MinD move from one pole to another in the cell in a pretty regular fashion. So just have to give it a little time here. Speed it up maybe a bit. And you can see now it's just beginning that the MinD will be at one of the cell primarily, and then it will start oscillating towards the other end. So I'll just kind of slow it down as it begins to do this in a more obvious manner. So now we can see it's pretty much all segregated in this half of the capsule, and then if we just give it a little bit more time, it will then oscillate to the other end. And so we can see this diffusion property really well in the system using VMD. we just freeze it here, you can kind of see that oscillation really well, how the majority of the MIND protein is segregated at this half of the cell, blocking the FTSC from polymerizing there. And so yeah, that's essentially what I wanted to show about how you can interact with the lattice micro simulations and visualize them in VMD. If anyone has any other questions, I'd be happy to take those. But I just wanted to show as well, if we go back to the presentation, the types of plots that we can get from this data that will show the oscillations really well is you can make a plot of the position of the MIND proteins over time and plot the axis position of the cell on the y-axis. And so you can clearly see again this oscillation from one end of the cell to the other on the order of about 30 seconds. And that's directly from our simulation file that we can make this plot. And if you're more interested in making movies in VMD in a little bit more professional way, I'd invite you to look at this tutorial that is on the center's website about making movies in VMD. And then finally, our newest software release of Lattice Microbes, 2.4, which should be coming out in the next month or two, has really excellent documentation. It's also available in AWS for you right now about how to do this type of visualization in VMD and how it will allow you to view different organelles and regions in the cell, how it lets you see the geometry, the actual lattice spacing of the cell, and then how it will let you see specific species and their dynamics throughout the cell. And so just as before I kind of wrap up this talk, I'd just like to acknowledge Professor Lucy Schulten, Tyler Ernest, who you've all met, for developing this plugin for lattice microbes in VMD, Mike for his development of lattice microbes, and then John Stone for helping with the integration into the overall VMD program. And kind of before I close or take any more questions, there's one thing I just realized that I kind of forgot to show, and we'll see if we can pull it up here. So when VMD is loaded, it actually starts a window in the terminal. And so that will also give you some pretty interesting information that you can look, such as the simulation time it will print out, how many frames you're going to look at, and then how many different species there are, and how many different site types there are, so cytoplasm, membrane, and so on and so forth. As well as the overall number of lattice sites that you have, so that's kind of given in the traditional VMD atoms representation, as well as the size or the dimensions of our lattice. Okay, so up here. And I guess that concludes everything that I wanted to say. So if anybody has any questions, I'd be happy to take those. Or if not, I think we're done.